Imagine a world where crop diseases could be predicted and prevented before they devastate entire harvests. That future might be closer than you think, thanks to a groundbreaking advancement in gene mapping. An international team of scientists has unveiled a revolutionary method to enhance the precision of gene mapping in complex organisms, marking a crucial step in the fight against fungal diseases that threaten global food security. But here's where it gets controversial. Despite the vast amounts of genetic data available, predicting genes in complex organisms has long been a stubborn challenge. Past attempts to map the genes of fungi responsible for crop diseases like Septoria leaf blotch, a major culprit behind wheat yield losses in Europe, have yielded inconsistent results. Studies have identified anywhere from 10,900 to 13,200 genes, yet only a third of these findings aligned across datasets. Why the discrepancy? And what does it mean for our ability to combat these diseases? To tackle this, researchers at Rothamsted Research, alongside collaborators from the Netherlands, Germany, the US, Australia, and the UK, developed a cutting-edge bioinformatics tool. This tool combines multiple gene prediction methods with real biological evidence from fungal RNA sequences, offering a more accurate and comprehensive view of the fungus's genetic structure. The updated analysis identified 13,414 high-confidence genes, shedding new light on how the fungus regulates its genes and revealing previously hidden genetic variants. Dr. Jason Rudd, who led Rothamsted's contribution, explained, "By analyzing each gene in detail, we were able to form a consensus on their most accurate structure, uncovering genes and variants that were previously undetected." This collaborative effort has resulted in a gold standard, open community reference resource for Septoria, poised to accelerate research into the pathogen's vulnerabilities. And this is the part most people miss. The study also uncovered new insights into how the pathogen's genes are structured and expressed, particularly in regions controlling their activation. This could revolutionize our understanding of how fungi adapt to different environments, potentially leading to more effective disease control strategies. But it raises a thought-provoking question. Could this level of genetic insight lead to ethical dilemmas, such as the potential for genetically modified crops or unintended ecological consequences? Dr. Rudd emphasized, accurate genome mapping is not just a scientific achievement. It's a critical tool in safeguarding global food security. By refining gene prediction techniques, we aim to monitor and combat fungal pathogens before they wreak havoc on crops. Meanwhile, the UK is taking practical steps forward with a series of Agriculture and Horticulture Development Board (AHDB)-funded pilot trials. These trials will test the effectiveness of biofungicides, a type of biopesticide, against Septoria tritici in winter wheat over 2 cropping years, 2025-2026 and 2026-2027. Biofungicides, commonly used in high-value horticultural markets, are regulated as plant protection products. However, their biological nature requires careful integration into crop protection programs. Sacha White, AHDB's lead crop protection scientist, noted, while biofungicides show promise, their efficacy in field conditions where factors like weather and disease progression play a role needs rigorous testing. The trials will focus on Septoria tritici due to its economic impact on UK winter wheat and the availability of biofungicide products with disease management potential. Here's where it gets even more intriguing. The trials will test biofungicides on 2 wheat varieties with different resistance levels to Septoria tritici as part of conventional fungicide programs. By varying fungicide doses, researchers aim to pinpoint when biofungicides begin contributing to disease control and yield gains. But will these findings be enough to convince farmers to adopt biofungicides over traditional methods? Led by a consortium including SRUC, ADAS, and NIAB, the trials build on AHDB's long-term fungicide performance research and leverage strong industry connections. The results will provide independent data on biofungicide performance, empowering arable farmers to make informed decisions. As we stand on the brink of this agricultural revolution, one question lingers. Are we ready to embrace these advancements, or will concerns about cost, accessibility, and ecological impact slow their adoption? Share your thoughts in the comments. Let's spark a conversation about the future of crop protection and global food security.